let's start then with the first of our 3 talks in the first session, and that's Paul Newton, Professor of Tropical Medicine, and as far as I know, one of the internationally key persons when it comes to researching about falsified medicines. So we are very much looking forward to your talk. And do you want me to say something more about you, or are you going to say something about yourself? Please, welcome.
Thank you very much. Thank you very much, Professor Absalund and Professor Linden, for the invitation to speak on poor quality medicines. So I'm based from Oxford, but actually work in Laos in Southeast Asia, and we're a part of a project to try and look at the global epidemiology and impact of poor quality medicines. So it's a forgotten history. Our ancestors were very bothered by this problem. The ancient Egyptians and the ancient Greeks were both bothered. An Egyptian princess led an expedition into what's now Ethiopia, 1500 BC, to find good quality medicines, because she was so bothered by the poor quality that she was then obtaining. The book on the left is a book about the trade in fake cinchona bark in the 1600s. Cinchona bark was the first, if you like, modern anti-malarial, and was very much sought after, and it was falsified on an enormous scale, such that people in South America and Europe lost confidence in it. And this is a book from the 1700s describing this and what they did to try to engage with people and encourage the use of it. In Victorian England, there were enormous problems with adulterated, falsified quinine, which is the compound in cinchona bark. And there were offices in colonial India and in colonial Vietnam directly concerned with trying to ensure that the quality of quinine in the 1920s and 1930s was good. At the bottom right, that's an illustration from 12th century Arabia, now Saudi Arabia, and it pictures physicians beating a pharmacist for selling them poor quality medicines. Many of you might have seen the film The Third Man, based in post-war Vienna, that's about fake penicillin smuggling. And that's Trevor Howard playing the British intelligence agent responsible for investigating this. Orson Welles was the criminal behind it all. And in front of him, those are vials of fake penicillin. And we've been trying to find out where this story came from. And we discovered that it was actually a true story, that in April 1946 in Berlin, there was an enormous seizure of fake penicillin, as this newspaper report describes. And it was being sold for £300 in 1946. I don't know what that's worth today, but it was such a valuable commodity that people were spending £300 on one vial of penicillin. So it was an obvious target for criminals, German and American, to make a fortune out of this. And it was discovered when one of them dropped a bottle of fake penicillin in a bar, and it was noticed, and that led to their arrest. So it's been a preoccupation of our ancestors, but it's been, I think, rather odd that over the last few decades we've rather forgotten about it, which I think is wrong and not evidence-based. Probably many of you in the room will be involved in all those medical specialities on the left that will then be used to determine optimum therapy for particular syndromes or diseases and informing national policy. But what's the point of that billions of hours of person investment and financial investment if the final common pathway of poor prescribing, poor adherence, and poor medicine quality are not good, so that they can be translated into the optimum efficacy and effectiveness for patients to use. So this final common pathway seems to have been rather neglected in the last few decades. So what do we mean by poor quality medicines? In our group, we use three different definitions. Substandard we regard as those resulting from factory error, from negligence, not deliberate, but the result of human error. On the contrary, falsified is the deliberate falsified production of poor quality medicine with the intent to deceive health workers and patients. Degraded medicines are made well, but because of poor, good pharmacy practice, etc., they're not stored or distributed appropriately, and they deteriorate through time. There have been reports of poor quality antimalarials that, as far as we're aware, except etovaguanol malarone. That's the only one that we've not been able to find any reports of either falsified or substandard examples. But if I was a criminal, I would be really keen to falsify malarone because it's so expensive, one can make quite a lot of money. So I think it's likely to be out there. One key example from the recent past is this large epidemic of malaria on the Pakistan-Afghan border in a refugee camp, 2003-04. This epidemic was very puzzling for those trying to deal with it. They first thought it was due to resistance to the antimalarial sulfadoxine pyrimethamine, or SP, that was being used to treat it. But there was no evidence of drug resistance. So then they looked at the content of the sulfadoxine pyrimethamine, and they found it contained exactly what it should contain. But they next did the crucial step that we often today do not do, in part because of its difficulty and expense, and that's to do dissolution testing as a surrogate marker for how it dissolves in the human gut. And that showed that it dissolved very poorly, so it would have led to very low drug levels in the blood, so the malaria parasites would have seen subtherapeutic concentrations. And there are anecdotal reports of sulfadoxine pyrimethamine in East Africa where people have eaten white tablets of SP, but they've come out the other end as white tablets because they've been compounded so badly. We first became interested in this during the large epidemic of fake artesunate starting in about 98, 99 in Southeast Asia. That was then, no longer, but back then that was the key antimalarial for the region. And one company, Gwilin Pharma, was targeted by criminals, and they produced enormous numbers of falsified artesunate. And they produced 16 different types of hologram and packet. It was like a Cold War between those investigating this and the counterfeiters. They made some silly mistakes, such as the spelling of tablet here, and there was one packet from Burma where the expiry date was before the manufacturing date. So that was a clear giveaway. But it was a major public health problem because many vulnerable people relied on oral artesunate at the time. But very little seemed to be happening. So a group of us teamed up with other organizations and actually looked to see what was in the tablets to say whether that would give a clue. The tablets were full of pollen. In Australia and New Zealand, there's a tradition of using pollen to solve crimes, and they demonstrated that the pollen within the tablets was consistent with an origin in southern China. Also, the type of chalk in it was very special. I thought that chalk was just chalk, but no, there are different types of chalk, and the chalk in these tablets was a type of chalk that's only found in southern China. We were very excited to find this beast on the right in one of the tablets, but it turns out to be an ordinary house dust mite, which would be the same as here in the floor in this lecture room. So the forensic analysis of this demonstrated that there were two main distribution routes of fake artesunate from southern China into Southeast Asia. And when the Secretary General of Interpol presented this evidence to the Ministry of Public Security in China, they acted very quickly, arrested people in China, and the Myanmar police arrested 3 in northern Myanmar. One of these packets was one of those seized by the Chinese police. The other one is a genuine example from the company. You have $1, you're a rice farmer in Laos, and you have to choose which one of these you will take. Would anybody like to hazard a guess which one you would take? It's very difficult. You can tell with a hand lens, but how many malaria patients carry hand lenses with them? Because you can look at the hologram and see that the hologram is fake. But there's one clue staring you in the face. And that's that. There's a dot at the end of tabs, which should not be there. So that's the fake. But it illustrates how difficult it is to tell the falsified from the genuine when the falsifiers do it well. This is another example that illustrates that not all falsified medicines do not contain any active ingredient. This was from the Democratic Republic of the Congo, and it did contain artesunate, subtherapeutic, and this would have engendered drug resistance. The French police went to the address at the bottom in Paris, but it was an ordinary house, and nobody in the house had any knowledge of AT-17 or pharmaceutical manufacturing. So presumably they'd just chosen an address at random. An additional thing that's become apparent is that many of us are involved with clinical trials, and the consort statement is a key part of that. But for both antimalarial and other clinical trials, there are no real guidelines to ensure that the quality of medicines used in these are good. And this is an example of one from Kenya, where the trial would have led potentially to erroneous results, and could have harmed patients. So we've recommended that the consort guidelines are changed to try and ensure that patients included in clinical trials are given quality-assured medicines. The WARN antimalarial quality surveyor is on the web, and in this we try and map all the publicly accessible reports of the quality of antimalarials. And this is the scheme for Asia. And as you can see, there's lots of red. Red is bad, bad quality, and there's not much green. But I would caution, because there's probably a lot more green there than we can see, because people tend not to report good news. People tend to report bad news. So this will be a biased picture, but I think it does, even bearing that in mind, demonstrate a severe problem. And the situation in Africa now is extremely dire, with many reports of falsified artemisinin-based combination therapy. There are many problems with the data available. It's relatively new science. There are many countries with no accessible data. Surveys have been done using convenience sampling, not using random sampling. There's been very little dissolution testing, and there's been little consensus over definitions. You don't need to see the details of the graph on the bottom right, but this is a graph of the number of samples taken in a survey on the x-axis and the number of surveys on the y-axis, and the red line is at 10. So the vast majority of surveys of medicine quality have had a sample size of 10, and they've used convenience sampling with no real sampling design or power calculation. So it's the equivalent of, would you agree with a study in Stockholm where you estimated the prevalence of hypertension in the city by walking around the city and the first 10 people you met measuring their blood pressure? I mean, that's a lot of medicine quality work is as bad as that. And there needs to be a great move to try to improve the sampling techniques for understanding the epidemiology of medicine quality. Impact, well, clearly if you have life-saving medicines and they don't contain what they're supposed to contain, if they contain wrong active ingredients, that's also quite common, it will have many consequences for patients, health systems, and the pharmaceutical industry, and also on drug resistance. On the right, this is a graph from WARN that shows this is real patients given one of the main antimalarials of today, artemether lumefantrine. On the x-axis is the parasitemia at admission, and on the y-axis is a measure of outcome, the recrudescence rate percentage at day 28. And there's declining dose as you go higher up. So the red line has the lowest milligram per kilogram dose, and the green line at the bottom has the highest dose. And as you can see, there's an enormous difference in the percentage of recrudescence of falciparum malaria if you have too low a dose, which is the case for substandard medicines and for some falsified. And on the left, that's a paper, the first evidence-based modeling of the consequences of poor quality antimalarials on outcome, on death, showing an estimated substantial number of under 5 children dying as a result of poor quality antimalarials based on the worn data. This will be wrong because as we've just been discussing, that the data are poor quality, but this will hopefully be refined as we have better quality information. And so drug resistance is a key thing. This is an example from Ghana of private sector falsified coartem that contained pyrimethamine. Pyrimethamine is the co-drug of sulfadoxine in SP, which is still important in Africa for the prophylaxis of malaria in pregnancy. And this is a very good copy. The main clue that you have when looking at it is that the spelling of the word store in German is wrong in the central circle. But again, how many malaria patients in Ghana read German? Probably not very many. So this was widely available in Ghana a few years ago. There's also, I think, a large problem in reporting. There was 1 million packets of public sector AM/FM coartem, as on the top left, were seized in Angola, and it took 5 months for this news to become publicly available through Facebook. And the head of the WHO Global Malaria Program heard about this through Facebook. The next report was in the Wall Street Journal 11 months later. So it's surely not right that this information is released, one, in an uncontrolled way without engagement, but two, how many malaria patients look at Facebook and read the Wall Street Journal. So as Panette will talk later about the WHO alerts, I think it's crucially important that we all report what we know about this to WHO and to the medicine and regulatory authorities of the countries concerned. Lots of things that we do not know that are crucial for understanding the problem and how to make the quality of medicines better. A key thing that I don't think we need any more research to understand is that with WHO estimating that 1/3 of medicine regulatory authorities globally have a, quote, no drug relation or a capacity that hardly functions. If we agree that medicine regulatory authorities are crucial for the regulation and policing so that we all have a good drug supply, that can't be right. And the international support for medicine regulatory authorities must be a key intervention. So thank you very much. You might have seen this poster that seems to become very popular, even though I gather it was actually never used in the UK. But I would argue in this case it's wrong and that we should not keep calm. Thank you very much.